project supported children and their families affected by cancer, pediatric cancer. And most of the funds raised over that time went to support the work of Camp Quality. The club also decided during that time that we should give some proportion of the funds raised towards a much more long-term goal involving research into paediatric cancer. And so in partnership with Australian Rotary Health and eventually the University of Sydney, the club endowed a PhD scholarship for research into paediatric cancer. The scholarship was awarded to Lesley Myo Thwe in, I think, 2011 or 10, 2011. Lesley is a graduate in medicine and a master's in pathology from Yangon, which used to be called Rangoon. She's been a GP. She's also been a pathologist. She also practices as a pathologist and has lectured in pathology in Yangon. Since coming to Australia, she tutored in pathology at the University of Sydney and before taking up her scholarship. Now, Lesley's now towards the end of her studies. She's travelled across from Sydney today to speak to us, and before I finally will invite her up here, I have to tell you that there's been a real glitch with the overheads today. The system is not working. We can't get the computer to talk to the projector, unfortunately. So Lesley has here all her overheads. It's in front of me at the moment. She'll talk to those, and you'll have to use your imagination. So without further ado, I invite Lesley to come forward and speak to us about her work.
Good afternoon, everyone. First of all, thank you for coming to my talk. Before I present my PhD project, I do have a few things to say. I am truly thankful all of you who continuously support me to do my project throughout my candidature. Support of your club means a lot to me, to the Australia, and to the world. I worked on the childhood cancer. It's a kind of rare cancer group, but it is important because all children are leaders in future. Our neuroblastoma project cannot be run smoothly without your support. Because of your support, we can have more understanding on neuroblastoma, and my results reveal the major significance to policies regarding biobanking and to many researchers internationally as well as within Australia who are working on biomarker analysis of rare cancers. Currently, neuroblastoma tissue microarray that I constructed was shared to the Children's Cancer Institute of Australia and to the Cancer Epigenetics Lab, University of Bristol in the United Kingdom for further research. My results were presented locally here in Australia and internationally in Spain at European Association of Cancer Research Conference and in America at American Association of Cancer Research. Detailed new protocols for laser capture microdissection and DNA extraction methods from formalin-fixed paraffin-embedded tissues are now available for researchers in our institute and beyond. So, your support is really meaningful for treatment innovation of neuroblastoma in future. On behalf of the Children Cancer Research Unit, my supervisor and associate supervisors, and myself want to say thank you very much, Australian Rotary Club of Athletes, from the bottom of our hearts, and we really, really appreciate it. Now I'm going to present my project. Sorry that I couldn't show you the PowerPoint that I prepared. The title of my project is Biomarker Analysis in Pediatric Tumours Diagnosed Within a Single Institution. The total proportion of childhood cancer represents approximately 0.6% of all cancers diagnosed. According to the data of Cancer Council Queensland, 10 years from 1997 to 2006, around 618 children are diagnosed with cancer each year in Australia, corresponding to around 156 per million children per year. And then I would like to show you that the incidence of the childhood cancer in statewide, but sorry. So first of all, we have to know what is neuroblastoma. It is most common solid tumor of infancy, and it involves mainly the adrenal gland, which is situated just above the kidney. Neuroblastoma is the leading single cause of cancer deaths of children under 5. And to date, we still don't know the exact causes. It occurs approximately 1 out of 100,000 children and more common in boys. Next, what are the clinical features of neuroblastoma? So as I present to you, neuroblastoma mainly involves in the adrenal gland, so it can present with abdominal mass, abdominal distension, pain, constipation, and even with enlarged liver and lymph nodes. It can also spread to any part of the body, so if it spreads to the bone, patient can present with bone pain and tenderness. If it involves to the respiratory system, lungs, it can manifest as a chronic cough and breathing difficulty. Neuroblastoma can spread to the brain, so if it spreads to the brain, patient will present with paralysis, inability to empty bladder, obsoclonus myoclonus syndrome, which consists of dancing eyes and dancing feet. And it can also spread to the eye, and we can see the recurrent eyes in the around the children's eye, and it can present with Honor syndrome, which consists of drooping of the eyelids, constriction of the pupils, retraction of the eyeballs, and absence of sweating on the affected side. And also, it will present with the vague symptoms like fever, loss of appetite, fatigue, pale, profuse sweating, rapid pulse, and high blood pressure. If the tumor not situated in the adrenal gland, it's situated close to the spinal cord and the vertebra, it will compress the spinal cord, and so patient can present with weakness, inability to crawl, inability to stand, inability to walk, or inability to empty the bladder, etc., etc. So mostly parents detect the symptoms. So next, we should know how can we diagnose the neuroblastoma. So there are many diagnostic tools. Among them, biopsy is the gold standard, and we can also do the imaging like CT scan, MRI, MIBG scan, PET scan, and also the biochemical tests like urine or serum catecholamines and its derivatives. And also, we need some supportive tests like a complete blood count. Neuroblastoma can undergo the pathways, if it undergoes the spontaneous regression or spontaneous maturation, which is really good, but it can progress aggressively. That's not good. Sorry, actually I want to show you the surgical resection of the neuroblastoma picture and then the histopathology pictures, but actually I want to say about the histopathology features that neuroblastoma we can see the neuroblastoma cells under the light microscope as a small blue round cells with the large hyperchromated dense nucleus with the scanty cytoplasm and increased in nuclear and cytoplasmic ratio, which is the features of the cancer cells, and also one of the histopathology features of the neuroblastoma, which is the rosette formation. The neuroblastoma cells are creating the rosette without the blood vessels. Sorry, actually I want to show you the pictures. So our hypothesis is storage time of samples affects the measurements of protein expression in pediatric tumor cohorts due to samples being required to be collected over long periods. Our objective is to examine whether biomarker expression can be validly compared in tissue samples of different ages, initially in the context of neuroblastoma. First of all, we collect the neuroblastoma cases diagnosed at our children's hospital at Westmead from 1985 to 2005, and we got 174 neuroblastoma cases. And then we proceed to construct the tissue microarray. So to construct the tissue microarray, we need to identify the cases that they have formally fixed paraffin embedded specimens, and they were approved for further analysis by histopathologists, and they were removed before chemotherapy commenced. So among the 174 neuroblastoma cases, we got only 56 cases to construct the tissue microarray. Then we collected the other clinical variables like patient stage, age, gender, patient stage, MYC amplification, chromosome 1 deletion, serum ferritin level, and the histopathological grading, favorable or unfavorable histology, and so and so. So, and we continue to analyze these both cohorts, overall cohort and analysis cohort. I would like to mention this. First, we collected the 174 cases, we call it overall cohort. And the 56 cases out of 174, we call it analysis cohort. So we use the analysis cohort cases to construct the tissue microarray. When we do the analysis, survival analysis on both cohorts, we found out that large proportion of the patients were in a stage 4 at the time of the diagnosis. So the higher the staging, the poorer the patient outcome. We have identified informative neuroblastoma patient cohorts when samples are collected over 20-year study period, and when we did the survival analysis, these two cohorts have very similar patterns and similar trends in stage of the disease, 5-year survival rate, MYC amplification, serum NSE level, and histological categories. Our analysis cohort includes also samples of different ages, which is 4.5 to 25.4 years old, and it is the same as sample age of overall cohort. So we can conclude that our analysis cohort, that is 56 cases, represents the overall cohort. Then we prepared to construct the tissue microarray. We collected the formally fixed paraffin embedded tissue and their corresponding slides and the data sheets, and we matched the slides and the blocks. Sorry, actually I want to show you how the steps of how to construct the tissue microarray using the machines. And using the 56 cases, I construct the new neuroblastoma TMA. What we call is analysis neuroblastoma TMA. It contains 47 neuroblastoma cases, providing the 82 cores of neuroblastoma tissue, together with the 17 cores of 10 control tissues. you will understand when I show you the pictures. Sorry. And we proceed to do the IHC staining using the automated BondMax machine and automated hemotoxin and eosin stainer. And we did the analysis of these IHC staining slides using the Aprio scan scope and TME lab version 9. We did the analysis digitally and visually. So digitally, we used the Aprio scan scope and TME lab version 9. And visually, under the microscope, we did the 4 visual scores, that is no staining, weak staining, moderate staining, and strong staining. And then we, depends on the staining expression, we did the IHC staining slides that comes from the analysis cohort. And we divided the specimen ages into 2 groups, the older specimen age and the younger specimen age. In our tumor bank, before I construct the neuroblastoma tissue microarray, there was a previously constructed archival tissue microarray in which the tissue cores were collected from 1942 to 2005, and it contains 112 cores of 56 neuroblastoma cases and 38 cores of 19 control tissues. But it had limited clinical information. For me, like the new neuroblastoma tissue microarray, it has a full clinical data. So we can do the full survival analysis, we can do the digital and visual analysis, IHC staining analysis on these 2 tissue microarrays. After the analysis, we can conclude that we did perform IHC staining on 2 different neuroblastoma tissue microarrays using 5 different biomarkers: neurospecific enolase, that is NSE, NB84, neuroblastoma protein 84, ALK, anaplastic lymphoma kinase marker, and tumor protein D52, that is TPD52, and MGMT, or methylguanine methyltransferase marker. And we found out that NB84 and NSE staining was significantly inversely associated with the specimen age in neuroblastoma samples collected from 1942 to 2005, as well as my neuroblastoma tissue microarray 1985 to 2005, respectively. So this indicates that the different antigens may degrade differently over time. So it supports our hypothesis. And that's why the sample storage time may significantly influence the results of IHC staining in neuroblastoma cohorts. Then we continue to do the isolation of the genomic DNA from neuroblastoma samples using the laser capture microdissection microscopy. Why we continue to do the isolation of the genomic DNA? Because we found out that the differential IHC staining was visually detected between the duplicate tissue cores of the same neuroblastoma cases in the ALK, anaplastic lymphoma kinase, MGMT, and TPD52 IHC staining. So we reviewed the whole section of these cases, and we got the 6 reproducible cases to continue to do the laser capture microdissection. So actually, I want to show you the laser capture microdissection machine. Sorry about that. And how we prepare the laser capture microdissection and how the laser capture microdissection machine works. Actually, I prepared for the video click, and then sorry, I couldn't show you. So after capturing the neuroblastoma cells using the laser capture microdissected machine, we continue to extract the DNA using the QIAMP DNA FFP tissue kit. So then extracted DNA from these neuroblastoma cases was quantified by a nanodrop spectrophotometer. And we got the total DNA yields varied from 0.12 to 4.05 picogram per micrometer squared. And then we wanted to know whether our extracted DNA can be PCR amplified using the D52 primers or the ORG primers. Yes, we can amplify our extracted genomic DNA by TP-D52 primers, but it doesn't work on the ORG primers. Actually, I want to show you the results slide. So after doing these tests, we can conclude that genomic DNA can be reliably extracted from archival microdissected formalin-fixed paraffin-embedded neuroblastoma tissues. D52 gene could be successfully PCR amplified. Downstream assays of DNA extracted from FFP samples may have particular design requirements. Extracted genomic DNA from FFP samples can be used for further research. So let me summarize. Survivor analysis and analysis of other clinical variables of both overall and analysis cohorts show similar findings that of other developed countries, and different antigens or proteins may degrade differently over time. Sample storage time may therefore significantly influence the results of immunohistochemical staining in the neuroblastoma cohort. Extracted genomic DNA from more than 20-year-old FFP sample can be used for further research, such as the treatment innovation. Last but not the least, I would like to acknowledge, sorry, I show all the people here, and special thanks to Australian Rotary Club of Adelaide for funding. Thank you.
Thank you, Lesley. Are there any questions? Thanks, Peter.
Thank you very much for an interesting presentation. To me, science and research sometimes is like dropping a stone into a pond. The ripples go out once you drop the stone. You seem to have dropped a very big stone in a particular area. You mentioned that towards the end that there are other impacts on research and other work. Could you mention or refer to any other cross work that's been done as a result of your activity?
Sorry, will you explain me the question again? Yeah.
Sorry.
Which works you want to know? Like?
You've done your research.
Yes.
Are you aware that other people have used your work to further expand your work?
Oh yes, actually it is. We got the contact with the Children Cancer Institute of Australia, and they are doing the slides from my neuroblastoma tissue microarray because it has full clinical data, and they will proceed to do the next generation. We call it the next gen, and they will find it out like how many genes were involved in to cause the neuroblastoma, and whether they have the overexpression or the mutation, and they also look at the other pathways. How can we block the neuroblastoma cells not to proliferate and to kill the neuroblastoma cells? Which pathway is the best one, or do we need to do like the many pathways to block it or something like that? This is the Children Cancer Institute of Australia, but I'm still waiting what is doing in UK, the University of Bristol. Yes, I'm still waiting how the results going on. Yes.
Thanks. Are there any? Yes. Thank you.
I'd like to congratulate you on a very difficult topic without slides.
Thank you.
I recognise some of that terminology, the my first patient as a nurse many years ago was 14 years old, dying with brain cancer. And I'd been a nurse for 6 weeks, so I didn't know much about anything back then, but I still remember the significant impact of that upon myself and the family of this 14-year-old boy who was unconscious when I was looking after him. I think we need to commend any research that's done for any child, any cancer in itself, but especially childhood cancer because of the impact. And I look forward to being able to see in the future the research that you're doing and your colleagues to actually help us to help those children more quickly and to improve their survival in the future. So thank you for what you're doing.
Thank you.
So I think we've pretty run out of time for questions, so it's my pleasure to present you with a little token remark
Thank you very
for speaking
much.
to us today, and ask the members to thank Lesley for her talk to us today.